And welcome back to the last lecture of season 16 during this self-isolation episode 10 of season 16. This is just a short video, focus on the body, basically taking what we learned in the last lecture about enzymes and narrowing in on some key enzymes in the body again. I know we kind of did that in the lecture. We talked about hexokinase and the other enzymes that are in the body too, but here's another way. It's a good lecture. I think you guys will like it. So basically, two different ways, and this is, you also like focus on the body and medicine is what these are called. So this is like how it's done in the hospital by doctors. I'm not a doctor, by the way, so excuse me if I misquote something. I know half your parents are doctors, and so you could probably call me on half this stuff, but in the hospital, one thing that doctors can do, or doctors will order the labs, is they can check for enzyme levels as a diagnostic. So what you can do is you can take a blood test and you can send that to the lab and you can test for enzyme levels in the blood, and boom, that's a diagnostic tool for you to figure out if something's going on in the body. Half the times we don't really test to see if something's present in the body because there's not really an easy way to do that. The easiest way to do that is to check for enzyme levels that will be like the red flag, if you will, or the green light that something's there, and causing those enzyme levels to skyrocket or to rise. And so that's a great diagnostic tool for people that know how to use it. Again, this is like more of a hospital type setting. The other thing that can be done with enzymes is if you want to treat somebody for treatment, what you can do is you can, instead of treating the actual thing that is in the body or the disease, the best mechanism or way about it for therapy for somebody is to interact. I think my chair just broke. Is to interact with enzymes. So if you interact with enzymes, then you're essentially going to be stopping chemical reactions from taking place. And so these are how enzymes are used as a diagnostic, or how they can be interacted with as far as treatment goes. So let's look at what the book had to say. So, for example, every cell is different, by the way. They all have some similarities, but each cell has different enzymes in it depending on what it's doing. And so sorry, I just checking a text message. When the cells are damaged by injury or they rupture and die, what they're going to do is release those enzymes into the bloodstream, and you can say, ooh, that must be a liver cell doing that, or that must be a kidney cell that's dying because you got a bunch of these enzymes in the bloodstream. And so here's a table of 4 enzymes. These are just 4 of many, but these are super common ones. For example, if someone's coming in with chest pain, you don't just immediately say they're having a heart attack, but you would immediately want to maybe do this creatine phosphokinase enzyme test, because it's probably pretty easy and simple to do, and that's a better guarantee that someone either is having or had a heart attack. Because if muscle cells die, then these enzymes will be elevated in the bloodstream. Okay. Again, I'm not a doctor, so I don't know if that's because of the rupturing of the muscle cells in cardiac tissue, and those enzymes are in those cardiac cells, or they're enzymes that work with cardiac cells, and if the cardiac cells aren't there, then the enzymes are going to be elevated because they don't get used. I don't know. But either way, there's going to be a direct relationship between dying heart cells, because that's what a heart attack is. It's just tissue death in the heart. Dying heart cells and elevated creatine phosphokinase enzymes. Now, same thing for liver and bone disease. If you have liver cells or bone cells that are dying off or have a disease or not working properly, you'll have alkaline phosphatase enzymes that are elevated. Prostate cancer will end up with acid phosphatase enzymes elevated. diseases of the pancreas will end up with elevated lipases and amylase enzymes, because those are what break. You already know, again, right? Amylase. What is that going to do? You remember amylose, starch? So amylase is going to break down starch. Lipase, remember lipids? So lipase is going to break down lipids. And so if you have these elevated enzymes, then something's going on. You must have a disease of the pancreas. And it might mean that they're not doing their job either if you have too much of them. Because they're not doing their job, they're just staying in the bloodstream for whatever reason. And again, so these are diagnostic tools, but just so you know, these elevated enzymes could cause a chain reaction and cause other problems, by the way. So, but anyways, that's all. It depends on the disease, depends on obviously the enzyme and all that. And so, but the point of 16.10a was that enzyme levels can be used as diagnostic tools. Okay, the second thing that they can be used for is treatment. You can treat diseases with drugs that interact with enzymes, like as inhibitors, remember? So inhibitors keep enzymes from doing what they need to do. A very popular one for HIV, the human immunodeficiency virus. We'll come to that one second. Let's do this one first. I'm sorry. I'm going backwards here. We're doing this example first. ACE inhibitors. Huge. These are huge in North America because a lot of people have high blood pressure. For whatever reason, whether you say it's diet or whatever, but North America has a really bad problem with high blood pressure. And so what you can do is, forget the reason, you can use ACE inhibitors. And they're a group of drugs that are going to do a very specific thing. And so what they're going to do is, so you have to follow my train of thought here, right? So angiotensin is a peptide, just a small peptide, not necessarily a protein. Actually, here it is right here. Angiotensin is a 8 amino acid long peptide. Angiotensin is a peptide that narrows blood vessels, and that increases your blood pressure, right? If you strengthen the diameter of a pipe, the pressure is going to go up. Okay, if you have the same amount of water running through a pipe, but yet you put your finger over the tip or you do whatever you can to narrow the pipe or the vessel, the pressure is going to go up. That's why when you put your finger over a water hose, it shoots out with higher pressure, even though you didn't really turn up the water. And so angiotensin does that. It constricts the blood vessels. Well, that's going to cause your blood pressure to go up. And so if first of all, here's how angiotensin is formed. It's formed by taking angiotensinogen, which is just 2 extra amino acids longer. This is the inactive form, basically, of this. And if you use an ACE, if you use the angiotensin converting enzyme, that's what ACE stands for. ACE is the angiotensin converting enzyme. So this enzyme will cleave this peptide bond here and release the histine and leucine. So the histine and leucine will just get released on their own, and boom, now you have it. Now you have this active form of angiotensin. Isn't that weird how just taking the same molecule here, the same polypeptide over here, but adding histine and leucine totally renders it inactive? Isn't that crazy? So, but if you have the angiotensin converting enzyme, the ACE, then it'll do that. The ACE will cleave that bond and get rid of the histine and leucine, giving you angiotensin. And that's going to increase your blood pressure because it's going to strengthen the diameter of your blood vessel. It's going to constrict it. And so by blocking the conversion of angiotensinogen to angiotensin, then your blood pressure is going to be decreased. It's not going to, or let's just say this, it's not going to increase. How about that? Which does mean decrease, but the point is you're not going to make it increase. So it's sort of an indirect way. But there's several effective ACE inhibitors. So remember, ACE is the enzyme that breaks that bond, but an ACE inhibitor will stop that from happening. So ACE inhibitors, you have, lisinopril, Zestril, Catepril, which goes by Capitan. I'm probably pronouncing these wrong. enalapril, which is pronounced Vasotec. These are generic names, trade names, whatever. We're not going to go into all that. But I will probably, the most important thing to note is this ACE enzyme, the angiotensin converting enzyme, ACE, and how an ACE inhibitor blocks that. And it might even want you to know about the histine and the leucine, since it's an open book test probably. And so last example, and then this lecture's over. Several enzyme inhibitors, and then I had something I wanted to tell you about that coronavirus assignment coming up. Several enzyme inhibitors are also available to treat HIV. So this is how these work. The most effective treatments are going to be the HIV protease inhibitors. And the way those work is, remember, we can analyze this term, protease. What does that mean? Well, ACE, it's an enzyme. And so protease, that means protein. So it's not like a lipase, which breaks down lipids, but it's a protease, which breaks down proteins. So the human immunodeficiency virus, or commonly known as HIV, that virus has a protease enzyme. And that protease enzyme is necessary for allowing it to reproduce and go infect other cells. Well, if you can inhibit that enzyme, then you prevent that virus's protease from doing its job and breaking down proteins. Because it's going to break down those proteins basically and then rebuild them, rebuild another protein with those amino acids. So it's kind of like this. Here's a good analogy. You got a kid who loves to build Legos, and you give him instead of giving him a Lego set and telling him to be creative and build something, you give him something already assembled and you say, okay, now break this up and now build something for me. Well, if you don't allow him to break up the Lego assembly that you gave him, then he has no way of building anything. That's kind of what's going on here. If you don't allow the HIV virus to do its breaking down, do its protease, if you don't allow protease to do its job of breaking down proteins, then it won't be able to build any more new proteins. In other words, it won't be able to make copies of itself and go and infect other cells. And so, Prenavir, Agenerase, maybe is the trade name. I hate drug names; they're so hard to pronounce. It's a protease inhibitor taken like twice daily by individuals who are HIV positive. Here's the three-dimensional structure. All right, notice all the carbons, hydrogens, nitrogens, oxygens, and every now and then a sulfur. Okay, so here's just a ball and stick model of the HIV protease enzyme. So you see how it looks like either this is the active site or that's the active site. I don't know. Oh, here it is. Ribbon diagram with the protease inhibitor Amprenavir in the active site. So that's what it does. The drug will go to the active site. And so that would be a competitive thing, wouldn't it? That would be a competitive. Now, if there's a covalent bond there too, it would be irreversible. But, okay, so that's it for this chapter. Now, what I wanted to mention about that coronavirus assignment, and again, I'll put a, wait, what's going on here? Sorry. I'll put a Google Classroom assignment up with a few more details of what I expect, but I'm not looking for like a 10-page essay. But if it takes that much to say what you want to say, that's awesome. And I don't mind if you borrow some material, but you got to do some citing and don't just copy and paste something. But you can use other, obviously, material and pictures. Just cite where you got the stuff from so you don't get in trouble, because we are an academic institution. But I'm not necessarily holding you guys accountable yet for the not MLA, that's for English, APA format. Although I should. That's my end goal. Each year now I'm going to start having, especially you guys, juniors and seniors, do APA style papers, but I know it's a little late for that and giving you resources and all that. So I don't want to hit you with that. But I would like you to cite your stuff so that we don't get into bad habits. And how you cite that, just try your best to do it APA if you haven't learned that already. I bet you guys have from Miss Gussie. But so just write something up that anything creative about, especially considering what we just talked about, how enzymes can be used as a diagnostic or how they can be used as a treatment mechanism by interfering with or interacting with those enzymes. But even if it doesn't have to do with that, if it has to do with anything about proteins, because this chapter is about proteins, and the coronavirus is known for that spike protein. But if you can find anything interesting about treatment of coronavirus, then that would be really awesome. If you could just write a short little summary, again, not a paper, not necessarily a 10-page paper, or even a 3-page paper, or even a 2-page paper. If you can do it in half a page to a page, that would be really cool. But I would like an introduction, like an abstract of just what you're talking about, and then show me what your stuff is, and then in conclusion, so I can like organize it. Don't just write like a letter like you'd be writing to a friend or a text message. But I would like it would be really cool if it had something to do with treatment of the coronavirus, but that might be a little trickier. So if you're just finding something about the mechanisms of how the proteins on the coronavirus interact with, again, or the history of, that's up to you. I want the assignment to be pretty open, but yet definitely tied to the coronavirus and the concept of proteins, and something that will be enjoyable for me to read, right? Because I find this stuff kind of interesting, and hopefully you will too as you do your research and find something that you can just kind of go down a rabbit hole with. All right, we'll see you later.